Hello, everyone. Good afternoon. We'll get started. It's an honor to introduce today's speaker, Dr. Luke O'Neill, who's visiting us from Trinity College, Dublin. Dr. O'Neill earned his undergraduate degree in biochemistry from Trinity College, Dublin, before completing his PhD in pharmacology at the University of London. He then conducted postdoctoral research at the Strangeways Research Laboratory in Cambridge. Today, he is a professor of biochemistry in the School of Biochemistry and Immunology at Trinity College, Dublin, which is a very beautiful campus I had the honor to visit. Dr. O'Neill's research has uncovered key molecular pathways that drive inflammation, including fundamental discoveries related to toll-like receptors, as well as TLR signaling, inflammasomes, and metabolic reprogramming in immunity. His work has been pivotal in understanding how the immune system responds to infections as well as inflammatory diseases. Importantly, he's also published multiple high-impact papers in journals such as Nature, Cell, and Science, and has also translated his discoveries into therapeutic applications, co-founding multiple successful biotech companies focused on immunometabolism. Dr. O'Neill's contributions have been widely recognized, earning him numerous prestigious awards. He was elected as a Fellow of the Royal Society in 2016. He is also a member of the European Molecular Biology Organization, or EMBO, and more recently, he was appointed to the governing body of the European Research Council. Beyond his scientific achievements, Dr. O'Neill is an exceptional science communicator, engaging in the public discourse through his best-selling books, as well as multiple radio and TV interviews. He has also awarded the Science Foundation of Ireland Scientific Communicator of the Year Award for his work during the COVID-19 pandemic. With his pioneering discoveries and dedication to both scientific progress and public engagement, Dr. O'Neill continues to shape the future of immunology and inflammation research. So it is an honor to have him here as our CTSI guest. And please join me in welcoming him to UCLA.
Thank you, Anthony, for those kind words. I wrote that introduction, by the way. You didn't mention enough of my awards. No, it's great to be here in UCLA. I haven't been in UCLA for about 15 years. It's remarkable. So it's nice to come back, see some old faces like Alex, who have known Alex for a number of years. Is Steve Bensinger here? There he is over there, that other one. So I do know some people here, which is quite nice. But great to come and give you a talk. Now, the beauty of this is I've got you here for 45 minutes. You're all trapped. The doors have been locked. Is this a compulsory course for some people? Is that why there's so many people here? It could be for credits. But it's great to have. Most conferences we go to, it's a pain because you got 10 minutes to give your talk or 20 minutes. You know, it can be difficult, especially for me because I never stop talking. And I've got 3 stories to tell you today. So it's great to have a bit of time to stretch my legs in this area. Now, the area, as Anthony says, is immunometabolism. And I hated metabolism all through undergraduate years. Learning the TCA cycle was a chore for me back then. And my main research was signaling, and I worked on TLR signaling. We discovered some of the adapters. We found bits of NF-kappa B, like Alex had done, and that was maybe 20 odd years ago, I suppose. And then 2012 was the road to Damascus, if you know what that is. I suddenly saw the medium go yellow with macrophages that we gave LPS to. And that had been seen 50 years before, of course, and it went really yellow. And I wondered, why is the medium going yellow? And it was lactate and glycolysis. And my lab suddenly began to shift into metabolism. And I must say, for any trainees here or young people, do that. Go down a road you don't like. And you might find awkward because there was a big difference from working on signaling into metabolism. And in 2012, I gave my very first talk on this at a conference. It was a Cytokine Society conference, actually. And nobody understood it because they're all immunologists. And afterwards, 2 very senior immunologists said, what are you doing? Glycolysis and Krebs cycle and macrophages, waste of time, you know. They were wrong because what happened then was this area of immunometabolism began to grow. And again, lesson number 2 is jump on a bandwagon early. It'll help you, you know. And suddenly many labs began to work on metabolic change in immune cells. And it's become extremely interesting and exciting for various reasons I'm going to tell you. Now, as Anthony said, I'm going to give you some translational stuff because as I've got older, I've decided instead of spending my time publishing fancy papers and swanning around the world, how about helping patients? What a radical idea. So luckily, 3 of my discoveries are in the clinic, and I'm going to give you that, mainly in phase 2 trials. And that's the biggest thrill. And again, a lesson for young trainees, keep going. Every paper counts, actually, because it contributes to a body of knowledge that might convince a company to go after a target. So never worry that your paper is in some journal that people don't rate. That's nonsense. I hate this publication racket, by the way. It's atrocious in our field what's happened, that we're all forced into these big journals. And if we're not publishing in those journals, we're somehow failing. That's complete nonsense. In my lab, if I see a JI paper, it's fantastic achievement. And if I get a CV off someone and they've got one JI paper and I read it and it's really interesting, that's as good as a big fancy paper. So never forget that. And as I say, every paper does contribute. The bulk, what's going to happen is, why would a drug company pick target X to go after? And target X can be anything. There's a bulk of literature behind that, and your paper could be in that bulk. So never forget your papers really count. And in my case, I'm more lucky, I suppose, because 3 of my papers gave rise to new therapeutics that are now in the clinic. And I'm going to give you some of that story as we go along. Now, the lab at the moment is obsessed with these things called mitochondria and how they drive inflammation. That's my title, very simply put. And we work on inflammatory diseases. Our main focus are these diseases ultimately. And these are staggeringly important. 1 in 3 people have some kind of inflammatory disease. The global cost, they reckon, I came across this a few weeks ago, is $47 trillion for all of these diseases combined. Now, remember, we're talking about the big ones like rheumatoid, MS. We're talking about colitis, Crohn's, psoriasis, atopic dermatitis, Parkinson's, Alzheimer's, atherosclerosis. These are all inflammatory. Probably 120 different diseases involve the inflammatory process going off kilter and being overactive. So it's a huge burden on humanity. And I always talk about this to my students. I still teach in the university. You're 22 years of age, you're a young woman, you begin getting diarrhea, you feel very awful cramps, and you're told you've got Crohn's disease. That's a very challenging diagnosis. Or maybe you're in your 50s and you begin to get movement problems and you go to your doctor, that's Parkinson's. So these are diseases that are extremely debilitating and very important. And I think with the way things are going, they're going to become more important than cancer. Now, cancer is obviously extremely important as a disease, but because of immunotherapy and various advances there, we're going to crack cancer. We're going to make it into a chronic disease, for instance. And then we'll see these other chronic diseases overtaking cancer, possibly, as the biggest burden on humanity. And of course, we will need to see that cancer sort of progress deliver. It is delivering. It needs to get a bit better. But it's very interesting to think about that. There's a huge number of these diseases and they're a huge burden on us, and that will continue to be the case. And even worse, there's an epidemic of them. And you may know this as well. There's a massive increase in autoimmune disease. This is in the Scientific American about a year and a half ago now. Autoimmunity has reached epidemic levels. Now, this isn't just diagnosis. There's something going on. And I'll come back to why I think it's the mitochondria to explain this, by the way. And the evidence is so compelling. Here's lupus. Over time, beginning in 1980 up to 2020, increasing incidence across all age groups in lupus. Here's rheumatoid arthritis, another very common one. Again, males and females, increasing over time. As you may know, many of these diseases are more common in females than males. 4 to 1 actually overall. With something like lupus is 9 to 1. Why that is, we've no idea. It's a really important question to try and answer. But the increase in incidence is a feature of all these diseases. One last one for you is MS in Europe has gone up 30% in the past 9 years. Now, this is far too fast to be genetic, remember. It's happening too quickly. It's got to be environmental. And we'll come back to what we think the environmental triggers for these things are. Now, I think the answer to the question as to why these things are increasing is to do with metabolism. And I'm going to make that case for you. And this field of immunometabolism is interesting. The term was coined by Diane Mathis. You may have heard Diane in Harvard. She was mainly working on what you'd call systemic immunometabolism. So before it got really exciting, in my view anyway, things like diabetes, obesity, they involve the immune system, and there'll be stuff on the immune system affecting these processes. Things like cachexia in the tumor microenvironment, all of those things were tied into immune cells at various levels. As you may know, in obesity, the visceral fat fills up with immune cells. Gamma deltas are in there, macrophages. It becomes like an immune organ in a way. So there's always been that going on, the systemic aspect. The second aspect is cellular, and that's the biochemical pathways inside the cell changing. And these two things kind of combine into this field of immunometabolism. Now, the cellular part, it is bioenergetics as ever. It's anabolism and catabolism. Now, that's classical biochemistry, you might say. But more excitingly for me and many of us, it's to do with metabolic reprogramming and the cell's phenotype. And what's happened is, very simply, and as ever in science, you need a new technique to help you. the microscope had to be invented, didn't it, to see bacteria? In this case, high resolution mass spec got better over the past 15 years, and you can see the metabolites. And they're changing dynamically. There's weird metabolites changing in different cell types. And what's happening is you can now phenotype an immune cell based on its metabolic profile. That's became very clear about 6-7 years ago. So in other words, this metabolic change is not just about bioenergetics or biosynthesis, it's also about the features of that specific cell and how that cell behaves in different circumstances. And then, of course, to translate it, the disease state, you'll see a metabolic change inside the cell there as well. So it's become a hugely fruitful area for immunology, because we can now explain, to some extent anyway, metabolic change in, say, an NK cell or a Treg or a Th17 cell. You will see differences in metabolism in those cells that govern the phenotype of that cell. In other words, if you intervene now and change that metabolism, the cell changes. You can turn a Th17 cell into a Treg by targeting metabolism. You can take an inflammatory macrophage and make it anti-inflammatory by blocking glycolysis. And that notion began to grow about 10 years ago, the capacity of targeting a cell in a way that you might want therapeutically. And that's the bit that I'm going to come back to in the translational aspect. And of course, it's very challenging, especially for immunologists who are not as smart as biochemists, right? I think. That's a joke. You can laugh if you like it. They're jokes. Half my phrase are there. Here's the metabolic pathways and here's the immune system. Look at this ridiculous, right? And recently, and I was telling Tim this earlier, actually, Eric Vivier, big NK cell lab, I met him at a conference about 18 months ago, and he said to me, we've got all this NK cell data, NK cell omics in humans, he said, and we think there's 3 types of NK cell in the human, and they're all different in terms of proteomics, they're different in terms of transcriptomics, and we think the metabolism is different, and they had all this metabolomic data. He said to me, would we help him interrogate that data? I made a mistake of saying yes, because 50 files arrived, my computer crashed. The trouble is, there's so much metabolomic data out there now, it's very hard to get into a soundbite, but the data came in, and Dylan Ryan, a very talented former graduate student of my lab, he's now in Cambridge, we went through it and we could help him. And you end up with this one little bit from the paper. So here we see the different NK subtypes, 1B, 1C, 3, 1A, and here's different metabolites, and there's big differences. So all that it's very descriptive, we can describe the different metabolic profile of, for example, an NK2 cell versus an NK1, and you'll see differences here. You see big differences in fatty acid metabolism, for instance. What this means, we've no idea, actually, but this is a treasure trove. Can you imagine now digging into that metabolic change and comparing the different NK cell subtypes and maybe then manipulating them? And this kind of gives you one little snapshot of what's happening. We're getting massive metabolomic data now from different cell types in the immune system, and we're trying to now figure out what that means and extend that into different contexts. So it's going to be a very interesting period, I think, to continue with this analysis and try to work it all out. Now, mitochondria are where it's at for me. That's the main thing I work on metabolically. We've done stuff on glycolysis and the pentose phosphate pathway and the side as well, but we relentlessly go back to mitochondria, because much of the changes we see in macrophages, that's our main cell type, by the way, the macrophage, is mitochondrial, and that's become a huge focus for the lab. Now, there's a picture of mitochondria, cartoon. I decided about a month ago at a conference on mitochondria to go back to the very first paper. I love history, you see. If you look at the history of things, you can understand them more, I find, especially in science. How were things discovered? And then what was the next question? This is the very first image of mitochondria with the electron microscope, and it's the famous George Palada. It's 1952. This was published. And what he says is, at that stage, if you look at the history of mitochondria, it's got loads of enzymatic units. They seem to constitute the main energy system. I mean, that was still being debated at the time. And thirdly, he says it's desirable to investigate the morphology of mitochondria at the high resolving power of the electron microscope. So it's the first ever EM of a mitochondria. And look what they look like. They're pretty ridiculous looking things. I mean, isn't this poor? This is a mitochondrion, right? Now, at the time, that was advanced, because the light microscope, there were blobs that were indistinct. Even more importantly, his best image in this paper is this, and even this is rubbish, right? But at the time, it was fantastic. He's looking at the inner mitochondrial membrane here. He notices these vaginations inside the mitochondria, the first time that we'd seen what's now known as the inner mitochondrial membrane. And he says this, he says, the internal ridges of the mitochondria appear as the most probable location of the postulated enzyme chains. They knew the electron transport chain was there. It was oxidizing stuff. They knew it was inside the mitochondria. And he says it must be in that inner membrane. And that was the first time that had been suggested. Now, why do I show you this? Well, fast forward to today, we've got great images of mitochondria. The confocal technology got massively better. And now, from my lab, here's mitochondria, right? We'd almost take the lights down to make it exciting, but still, you get a feel for it. This is a macrophage, given LPS, right? Now, we use LPS a lot in my lab. It's the hammer blow to the macrophage. It drives the macrophage into a massive activation state. Everything's always very binary for me. Control, LPS. I made a living out of that, right? I've got 4 Nature papers of adding LPS to macrophages. How stupid is that? But still, it's the 2 states. And what's happened here is those mitochondria have broken up. It's called fission. And they form little blebs. This is after 24 hours. Early in the time course, mitochondria actually improve with LPS. But over time, they break up into these mitochondrial blebs. These are very toxic. They're releasing reactive oxygen. They're releasing their nucleic acids, which, as we'll see in a few minutes, is very damaging for the cell, right? So here we have a very damaged mitochondria undergoing fission. Now, on this side of the diagram here, look at these mitochondria. The difference between this and this is we've given these macrophages a compound called TEP46. Now, what is TEP46? TEP46 targets an enzyme called PKM2. PKM2 is in glycolysis. And if you add TEP46, it forms a tetramer. I know that's very complicated. I'll come back to it. One of my big stories. But if you tetramerize an enzyme in glycolysis, the mitochondria are fantastically healthy. They fused, huge, long mitochondria. Look at this, beautiful all together. We love these images because all these long mitochondria, these are aspiring normally. They're not releasing as much ROS, and they aren't releasing their nucleic acids. And all I've done here is add a small molecule that targets an enzyme in glycolysis called PKM2. Now, this state here is very anti-inflammatory. Those macrophages are making IL-10, for instance, by the bucket. And IL-10 is a very important anti-inflammatory cytokine. These are ones that are making loads of TNF, right? So in other words, you get the message, mitochondria, they shatter with LPS. A tiny small molecule can stop that. And now the mitochondria fuse and they're healthy and they're anti-inflammatory. And that's become a big theme of this whole area, really, of these 2 states, if you will. Now, what's also important is these have seen LPS as well, remember. And they're reading the LPS differently, if I can put it that way. They're reading it such that the macrophage doesn't undergo damage in terms of its mitochondrial function and begins to make different cytokines. And that becomes a theme as well, as you see as we go along. Now, I want to give you that story in more detail with 2 others, right? So there are 3 stories I want to tell you. The first is this PKM2 I mentioned, adenosine. You get more on that in a minute, and IL-10. A second story is complex 3 in the mitochondrial electron transport chain. And again, that links into IL-10. These are 2 IL-10 stories. And of course, for the non-immunologists here, IL-10 is a very important cytokine. It suppresses inflammation. It drives tolerance. It's a really important anti-inflammatory process. And my third and biggest story is itaconate, right? A metabolite that the mitochondria make, and that's anti-inflammatory. There are our 3 stories. Now, the doors are definitely locked because you're all going, I don't want to hear these stories. But you will at the end, watch. So first one, PKM2. Now, it's a collaboration between my lab and a lab in the University of San Paolo. 2 postdocs in my lab, Juliana Toller and Eva Paulson. They did all this work. And here's the 2 labs. We made a flag for St. Patrick's Day, actually, with the Irish flag and the Brazilian flag. And it's Jose Filo, Zico. It's his lab and my lab. His lab was a key part of this. So I want to acknowledge them up front. Now, what is PKM2? Let's start with that. There's glycolysis. You all know glycolysis, I hope. There's the last step. Peptipyruvate is PKM2. That will go to the mitochondria for oxphos or get made into lactate, as I hope you know. And there's the enzyme I want to tell you about, this PKM2 enzyme. It's a very interesting enzyme biochemically. Way back in the 70s, this was shown. In fact, another thing that's happening is we're rediscovering all the biochemical literature from the 60s and 70s on these enzymes and applying them into the immune system. There's a whole treasure trove of data already. It was known early on with this enzyme, it exists in 2 states. It can be a tetramer or a dimer, and that oscillates between the 2, right? And the tetramer is the one that's active. That's the one in glycolysis. That's the one that converts peptipyruvate, this tetramer, right? The dimer is a lot less active. And allosterically, that can be controlled. Various things like fructose 2,6-bisphosphate, which is in glycolysis, will kick it into a tetramer to enhance glycolysis, a feed-forward loop. Now, the biochemists spent decades working on the control of these pathways. But that control of the enzyme activity is all down to allosteric regulation. And that's been known for 30 or 40 years. But very importantly, a paper about 10 years ago now showed the dimer, the dimeric form, can go to the nucleus. This is a separate role entirely from its role in glycolysis. And when it's in the nucleus, it interacts with HIF, the transcription factor HIF, and it will regulate lots of HIF-dependent genes. That have been shown, first of all, by Matt van der Heijden at the NCI in tumors. Tumors do this, and HIF needs this dimeric PKM2. My lab then saw Matt van der Heijden's stuff. I met him at a conference. Inevitably, this often happens at conferences. And he said to me, we've got this compound that can stop that dimer and force a tetramer, right? And that compound is called TEP46. It will force this into a tetramer. And he had shown that has anti-tumor properties because it stops this dimer going to the nucleus to control HIF-dependent genes that are pro-tumor, if you can follow that logic, right? And he developed it as an anti-tumor compound. We then try his compound. And again, to cut a long story short, we found in the LPS-activated macrophage, that dimerizes, the PKM2 becomes a dimer, goes to the nucleus and regulates IL-1. And IL-1 turns out to be a HIF-dependent gene. Secondly, we tried Th17 cells. Same thing again. IL-17 needs dimeric PKM2. And in fact, if you add the tetramerizer, stops all this. And we could show TEP46 could block IL-1 and block IL-17. And of course, we thought that's a very interesting property in the molecule. Because if you block IL-1 in macrophages and you block IL-17 and Th17s, that'll be anti-inflammatory. And with the IL-17 stuff, especially for immunologists who are here we see TEP46 suppressing IL-17 production. It suppresses TNF, which Th17s make, and it suppresses gamma. This is a really good way, if you like, to stop Th17 cells. We went in vivo. EAE is a standard Th17 model. You may have heard of EAE, I hope, a demyelinating model. And these mice will develop demyelination, mean score here. Give them TEP46, it suppresses that demyelination. We think it's doing it by blocking IL-17 and IL-1. And so it worked in vivo as well. Now, that was all exciting. And we published that about 3 or 4 years ago now, I guess. But the second thing we saw suppression of IL-1, but we also saw a kick in TNF. And in fact, you get a huge boost in TNF production. Or sorry, IL-10. We saw a suppression of IL-1 and a boost in IL-10. And here we see massive IL-10 coming out of the macrophages in response to LPS. And we wondered, what's the mechanism of this? Now, we confirmed this again in vivo with Salmonella. So this is IL-10 in the systemic circulation of a mouse infected with Salmonella. And again, a big boost in IL-10 production and suppression of IL-1. Now, remember, that suppression of IL-1 is happening because you're forcing the tetramer. Remember that? And the dimer isn't there. But we saw this big kick in IL-10 in vivo as well. And of course, we wondered, what's the mechanism of that? Now, what was nice was TEP46, then we published that paper, and lots of labs mercifully pick up on it. And that compound, TEP46, is anti-inflammatory in all these models, all these preclinical models that involve macrophages or Th17 cells. So sepsis, collagen-induced arthritis here, for example, IBD models, cystic fibrosis, asthma, EAE, we've shown ourselves. All these diseases were treatable, if you will, in mice with TEP46. Now, of course, it makes sense. If this molecule can boost IL-10, that'll be immunosuppressive. And if it can suppress IL-1 and IL-17, that'll be anti-inflammatory. But the question we had was, how can we explain the increase in IL-10? Now, again, remember, again for the trainees, if you're a scientist, you're never happy, are you? Hand on heart, especially nowadays. What I mean by that is you're always asking questions. And there was unrelenting question in my mind. How will this boost in IL-10 be happening? We got the explanation kind of for the IL-1 bit, the dimer tetramer, that was good. What about IL-10? And then it got pretty complicated. First of all, we were able to confirm everything. And Zika, this guy in Brazil, he began working on this, having seen our paper. And he could confirm everything we showed. So TEP46 is boosting IL-10 here. Over the LPS alone, for instance, here's a time course. Again, a nice boost in IL-10. We knocked out PKM2 because it was a small molecule, and you suppress IL-10 by knocking out PKM2. So we knew it was going through PKM2. And then Zika then realized, well, look, if you're boosting glycolysis, now remember that bit, the tetramer increases flux through glycolysis. You should increase ATP production. And lo and behold, we did. Here's ATP being made. Put in TEP46, a lot more ATP is coming out. And this is extracellular ATP. We could measure outside the macrophages. And then we measured the role of a channel called the pannexin-1 channel that exports ATP. That repressed IL-10. Here's the knockout over here. suppressing IL-10 production. And here we see a small molecule, again suppressing IL-10. So this channel is needed to export the ATP. And then we knew about this. There was a literature that adenosine can drive IL-10. That was already published. So we wondered, was adenosine part of this? Now what happens here is the ATP comes out, gets hydrolyzed by CD39 into ADP AMP, and then CD73 makes adenosine, and there's an A2R receptor. And it turns out all of these were needed for IL-10. So here we see an increase in adenosine. So we can confirm the adenosine was being made. If you inhibit CD39, you get no adenosine being made. And look at IL-10. So the adenosine receptor knockout couldn't make IL-10 here. We use an antagonist that could repress it here. We had a CD39 inhibitor that repressed IL-10, and a CD73 inhibitor could repress IL-10. So all of these steps were needed then to see the increase in IL-10 production. And there's a literature on adenosine going back a good while, actually. Adenosine is well known as anti-inflammatory. It gets made during inflammation. The receptors go up, it gets released, and then it suppresses various events to stop cytokine production, partly through IL-10. This all began to make sense, that the increase in IL-10 was through adenosine. And then we looked at OXPHOS. Now, this is something that we often measure in our macrophages. And we had shown this first, I suppose, about 10 years ago now. LPS represses OXPHOS. Look at this panel here in the middle, just to make it simple. There's the control, there's with LPS. You can repress that. That's because the mitochondria have undergone fission, and there are all those damaged mitochondria. If you put in TEP46, you stop that. So TEP46 can prevent that decrease in OXPHOS, and we've seen that. And then, again, I've shown you this already. The mitochondria are wonderful. They're the LPS ones. They've got TEP46. So these are respiring normally. They're making ATP if you put TEP46 on, right? And then, of course, we wondered, was this to do with IL-10? Now, remember, the TEP46 is boosting IL-10. There was a literature that IL-10 is very good at protecting mitochondria. And Russell and Medzatov had a paper on this initially, that IL-10 can target metabolism and sustain mitochondria. And a lab in Dublin, actually, Claire McCoy, could show that IL-10 promotes OXPHOS through arginase 2. So we wondered, is the effect on OXPHOS, the boost in OXPHOS, due to IL-10? And indeed, that's the case. So here again, we see our OXPHOS measurements here, where you get repression of OXPHOS with LPS, right? Very clearly knocks it down. The TEP46 can restore it. That's the green line. But look at this. If you block IL-10 with an antibody, no OXPHOS. You really repress all the OXPHOS by neutralizing IL-10. So what does this mean? We think, surprisingly to some extent, the tetrameриc form of this enzyme, PKM2, will increase ATP production from glycolysis. That gets released, gets turned into adenosine, and that now drives IL-10, and that now boosts mitochondrial health. So again, amazingly, a very simple small molecule, these PKM2 tetramerizers, could do all this by targeting PKM2 and allow the macrophage now to make loads of IL-10, which now feeds back and protects the mitochondria. Now, of course, what this means is we have a molecule now that's very interesting. We can block IL-1 and IL-17 with the tetramerizer. Secondly, we can boost the IL-10 by forcing the tetramer. What would you do with that molecule? Well, my company Citrix, which I founded in 2017 with Jeff Rathmel, you may have heard of Jeff, another pioneering immunometabolism lab, Mike Rosenblum, T-Reg metabolism lab, John Powell. We all formed this company back in 2017, and they took our discovery, Citrix, and they said, what we'll do is we'll make a drug out of this. And they took TEP46, they made a drug-like molecule from that molecule. They've got fantastic structural data, tetramerizers, PKM2. Here it is, forcing the tetramer. And that's their lead compound. It's called 65219. Inhibits IL-1, inhibits IL-17, and boosts IL-10. And they said, let's go to the clinic. They went to a phase 1 trial. It was safe. Now, which disease would you pick? They've picked atopic dermatitis. And the trial in this disease is about to start. It started actually already. They're giving this PKM2 tetramerizer to patients with this disease. Now, there is dupilumab. You may have heard of that. That works a bit. It's only for severe cases. There's a desperate need from mild to moderate atopic dermatitis with a small molecule. And they've got loads of preclinical data. It's an easy trial to do. You can take tape strips. You can look at progress, you can measure skin. So that's what I've gone for this. Now, this could work in many diseases, of course, we'll see. But this is going to be the first proof of concept. And again, you can imagine how delighted we are with this. Because all this PKM2 stuff starts in my lab at a PhD student, and then we get these papers on the cytokines. Citrix get on board and make it into a therapeutic. That's our first story, PKM2. I'll come back at the end now to wrap it into the whole story in a minute. Now, second story. If you thought that was good, get a load of this next one. I'm going to ramp it up. It's like a gig. There's going to be a final encore. Second story is complex 3. Now, again, it's IL-10. Now, Nav Chandel, a guy in Chicago, you may have heard of Nav, he's a pioneering immunometabolism lab. He published a paper that complex 3, of all the complexes in the electron transport chain, who would have thought it, is essential for Tregs. If you knock out complex 3, you get no Tregs. And I'd seen that paper, and that was interesting anyway. It's interesting in the back of your mind. What would complex 3 be doing there, right? And then very importantly, about another conference about 5 years ago, I met a guy called Martin Brand. Now, Martin Brand was in the Book Institute. He's from the UK. I've known him for years, actually. He did a PhD with Peter Mitchell, the most famous mitochondrial guy ever. Martin had these compounds called sequels, and Anthony worked on some of these strains, as I learned. And what Martin had done was he was developing inhibitors of the electron transport chain to try and block different complexes, you know. And there are inhibitors. Mixothiazole is one that's used. That's a bog standard complex 3 inhibitor. But Martin made a compound called Sequel-3. Sequel-3 targets complex 3, but it blocks ROS production. Now, complex 3 is a great source of hydrogen peroxide and ROS. In fact, it's the main source of ROS into the cytosol, because complex 3 can release the ROS into the cytosol. And Martin made a molecule that would block the ROS, but wouldn't affect forward electron transport. It didn't block OXPHOS. It just blocked ROS. And that's a great tool, because if you block OXPHOS, you may have other effects, you know. And he said to me, would we try these compounds in my lab on macrophages? Now, it turns out Anthony was trying them as well, right? But the thing is, we stuck these on macrophages and did loads of experiments. And this is how Martin got them. He began with over 600,000 molecules, by the way, went through a big screening process, got to Sequel-3 in the end. So it's very elaborate how we discovered these compounds. And we got the compounds. And first of all, we showed they could block ROS. Here's LPS driving reactive oxygen. There's Sequel-3. So Sequel-3 can block ROS. Mitotempo is a controller. So again, we confirmed they could block ROS production. But then, to our delight, it got more interesting. We did a big RNA-seq analysis, as we all do nowadays. We stuck the data into some big data set and said which cytokines are being regulated, and IL-10 came out. And it turns out of all the cytokines, IL-10 is repressed. Red means repression. No other cytokines really. Here's mixothiazole, the bog standard complex 3 inhibitor. So we could link complex 3 into IL-10 production. We confirmed it then, measuring IL-10 directly, a suppression of IL-10. And you get a boost in TNF because there's a feedback loop in macrophages. IL-10 will repress TNF in macrophages. So if you block IL-10, you often see a kick in TNF. And that was more evidence, I guess, is the way to think of it. And then we did lots more transcriptomics, as we do nowadays. AP1 was a target. We think there was repression of FOS. And many AP1 dependent genes were down if you block complex 3. And guess what? IL-10 is AP1 dependent. So we think it's tied into AP1. We're doing a bit more work on that. And then we go in vivo. Now, we decided to go to a B16 melanoma model here. Initially, we used LPS, actually. That's LPS driving IL-10. Sequel-3 blocked IL-10 in response to LPS. And then we went to B16 because this is a great model for IL-10. Because it turns out IL-10 is pro-tumor in this model. IL-10 is immunosuppressive. It takes out the CD8s. It blocks the macrophages, and you get a pro-tumor response with IL-10. And there have been papers blocking IL-10 with antibodies or in knockouts, boosting anti-tumor immunity in this model. So we went to this model because it was IL-10 dependent. And CPG is used, actually, as the adjuvant in this model. And again, we check with CPG. Here we see Sequel-3 blocking IL-10 in response to CPG very nicely. There's a message and there's protein. So we confirmed you could block CPG as well as LPS, just to be on the safe side. And then the sun came out, and Alessia Zota in my lab, the PhD student, when she showed me this data, it's 2 years ago now, we got the first in vivo data. It was incredible. Massive anti-tumor effect in this model. So here we see survival. Now, the untreated mice, they're all dead by day 9. If you give them Sequel-3 on its own, you can stretch out survival. If you give them CPG, that's known to work, but the combination was remarkable. Got a massive survival here. Here's the tumor mass. So the tumor grows and grows and grows in the control. You give them CPG or Sequel-3, that has less tumor volume. The combination, no tumor growth at all. So we think this is happening because we're repressing IL-10 with Sequel-3. And then we're boosting anti-tumor immunity. We went to the immune compartment, measured loads of parameters here. I'll just home in on IL-10 positive macrophages are being suppressed here with Sequel-3. MHC class 2 is up if you give Sequel-3, and various other markers here. So what we think is happening is this then. TOL4, we use LPS. Complex 3 makes ROS. ROS drives AP1 to drive IL-10. If you block complex 3 derived ROS, now remember, these compounds are very specific. They're just blocking ROS coming off complex 3. You block the ROS, you block AP1, and you suppress IL-10. And in this context, you'll boost anti-tumor immunity because you're losing that immunosuppressive effect. And then about another year ago, I'm always away at conferences. They're very useful. I bump into Nav, the Treg guy. I tell him our data. He goes, Luke, you wouldn't believe it. I said, what? The complex 3 knockout macrophages can't make IL-10. Now remember, we had a small molecule. And I said to Nav, let's put these two together. Because our paper is all the small molecules. So if you've got the knockout agreeing, the beautiful thing is the complex 3 knockout macrophages can't make IL-10. And you know what he said to me? My postdoc needs a publication to be first author. And I'm going, well, we can asterisk them. No, so the postdoc refused. Isn't that sad? In the end, they've come out together back to back in Science Advances. Nav's paper on the complex 3 generated ROS for IL-10 using knockouts and our data with the small molecule. So it's probably true, is what I would say there, because there's a small molecule and a knockout in combination. Now, so there's a story for you with complex 3 and IL-10. Now, lastly, my third big story, I'll be back at the end to put them two together. So don't go away anywhere. Itaconate. Right, now what the hell is itaconate? Well, I sometimes give a single talk on this. It's the metabolite that keeps on giving is the phrase I use. There it is. This molecule is remarkably simple, and yet there's about 500 papers now on that molecule in the immune system. We published our first paper in 2018. There were 3 papers on it. It's remarkable that this has taken off about the role of itaconate in immunity. Now, what is it? Well, it's in the mitochondria. It comes off Krebs cycle. Aconatate is turned into itaconate by an enzyme called ACOD1. ACOD1 is a conatate decarboxylase, and this had been ignored for, I'd say 100 years. It was first described by Krebs himself in the 1920s. He tried to fit it into Krebs cycle initially. It didn't fit in, right? It's a byproduct coming off Krebs cycle. And to cut a long story short, it's anti-inflammatory. I'm going to give you that story. Now, we'd worked on succinate, that's pro-inflammatory. And we worked on fumarate, which I'll come back to. That's more immunomodulatory. But this one, this metabolite is anti-inflammatory, and that's the story I want to tell you. Did a big review on this if you're interested in Nature Metabolism on metabolic messengers. That was back in September. That summarizes all the literature on this particular molecule. Now, what does it do? Well, first of all, it goes through the roof in macrophages. We had seen it in 2016 as a very abundant metabolite in LPS-activated macrophages. Didn't know what it did. And then 2 labs showed one thing it does is it blocks SDH. Now, SDH is in the electron transport chain. It turns succinate into fumarate. And itaconate, a well-known SDH inhibitor actually from the 1940s, we had shown SDH is needed for IL-1 production. A thing called reverse electron transport happens. I won't go into it. But the bottom line from this was itaconate blocks SDH and lowers IL-1. And that was the first hint that this was going to be anti-inflammatory. But then my lab showed this. It turns out to be cysteine reactive. So itaconate will modify cysteines on target proteins, and they're very often inflammatory proteins. And then we made a derivative called 4-octyl itaconate, and that will mimic itaconate to some extent. There are differences, but there's some mimicry. That turned out to modify cysteines even better. And then dimethyl itaconate is used. These are 2 compounds. Bottom line, they all modify cysteine. When you look at what proteins are being modified, it's startling. It's NLRP3, Gasdermin D, TET2, JAK1, KEAP1 gets modified for NRF2. All of these proteins we've shown to be modified. They're inclined to be inflammatory, and then the modification inhibits them. So the anti-inflammatory effect is partly down to cysteine modification, and then you get decreased inflammation. Now, very importantly, a drug called DMF. Now, DMF is dimethyl fumarate. It's also called Tecfidera. Biogen had that in MS. Biogen were making $3 billion a year from this drug. You can make it for 2 cents. It's the biggest markup ever on a drug. But still, what does DMF do? It modifies cysteines as well. And we think DMF is mimicking the natural metabolite. Now, DMF was discovered in the 1970s to be anti-inflammatory, but no idea of a mechanism. We now think it's modifying similar cysteines to the natural metabolite itaconate. So it's leveraging a natural process is the way to think of it. So we're in the world of cysteine modification. Most recently with Eli Lilly, who we collaborate with, IRAK4 is a great target actually. There's a key cysteine in IRAK4. Itaconate modifies it and inhibits IRAK4. Now there's a great target because IRAK4 is used by all the TLRs and IL-1 and IL-18. So again, you can see now the literature begins to grow on this molecule being anti-inflammatory. Now there's also stuff for those interested in cancer. There's been a slew of papers recently being pro-tumor. So for example, it will take out CD8s. Itaconate will repress CD8 T cells, and you will see a pro-tumor effect there. And then there's a good review on this. It's found this disease. Frank Pestler has said that. And look at this. In all these cancers, ACOD is pro-tumor, glioma, lung cancer, hepatocellular carcinoma, melanoma. And again, it seems as if itaconate then is pro-tumor for various reasons. If there's lots of stuff on this in the past year. High expressing tumors, poor prognosis in melanoma. The levels in lung cancer correlate with progression. And then if you can enhance checkpoint blockade in the ACOD deficient mice. So mice that can't make it, checkpoint inhibitors work even better. So the cancer people are very interested in this pathway as one little bit. We don't work on that ourselves. Now, back to inflammation. We made that molecule 4OI, the derivative. Now we made it, Richard Hartley, our chemist in Glasgow, made it. He made it to mimic itaconate. It's got similar electrophilic properties, right? There are differences, as I mentioned. We gave it out to many labs. We published this in 2018, right? We gave it out to many labs because you couldn't buy it. And look at this. It's active in over 50 models of inflammation. Osteo, lupus, fibrosis, psoriasis, IBD, Parkinson's, Alzheimer's, etc. If you give mice that molecule, it blocks inflammation in all those models. Again, a very simple molecule based on endogenous itaconate. And we showed a very important target, NLRP3. There's a key cysteine here that gets modified and blocks NEX7 recruitment. So one reason why 4OI is anti-inflammatory, it targets NLRP3, which I hope you've heard of. Very important inflammatory pathway in macrophages. And look at this. This is recent. These two papers came out in the past year that blew my mind. Now, you got to watch me. I'm a zealot for this. So believe half of what I tell you. But there are these two papers. Exercise, you may know this, is good for you. And in particular, exercise promotes tissue repair. It's a well-known thing. And in a model of that, in mice, the injured liver, if you stick mice on a treadmill post-liver injury, the repair is quicker. That's been known for about 30 years. Guess what? It's all itaconate. If you knock out ACOD, they don't repair as quickly. Even more excitingly, steroids, anti-inflammatory steroids go through itaconate to be anti-inflammatory. And that was Gerhard Kroenke's paper last year in Nature, to give him full credit. And look at this now. This is some of the evidence in this. And remember, what he's claiming here is remarkable. As you all know, I hope, steroids are a mainstay anti-inflammatory. They save lives. They're very good acutely. Long-term use, no good because of side effects, right? But they're very important clinically. Look at this. This is a model of arthritis in mice. They get more and more inflamed. You give them steroids, all the inflammation goes away, but not in the ACOD knockout. If you can't make itaconate, you become steroid resistant. And there's more evidence in that paper to support this notion. And then look at this. Many people have seen these papers, and things move so fast now. In the past 18 months, 4 papers, different formulations. This could be a therapeutic, obviously. Nanoparticles, hydrogels, orally active products, and topical form as well. In models, all being anti-inflammatory. So big excitement about leveraging this molecule now. And what if an itaconate derivative could retain the anti-inflammatory power of glucocorticoids, but with less side effects? That's the holy grail for steroid people, of course. Maybe this is the bit of steroid you want. This could mediate the anti-inflammatory effects of steroids. Now, one last bit I want to show you, recent stuff from my lab on this that we think is important. It's the connection from itaconate into the inflammatory process. Fumarate, I'd mentioned that briefly, that goes up in macrophages. It's got many targets. We published about 1.5 years ago this paper. Now, by the way, there's a metabolomic screen in human monocytes. Look at this. There's itaconate, 6 log increase, incredibly. But we also saw fumarate, 1 log, right? Here's an increase in fumarate. Fumarate accumulates in the macrophage. We mapped the biochemical pathway just to make it even more complicated for you. Fumarate hydratase, that's the enzyme that turns fumarate to malate. That gets repressed in the macrophage. So fumarate goes, FH goes down. Here is the FH being repressed. Secondly, a shunt called the argininosuccinate shunt goes up to make fumarate. And these two effects give rise to fumarate accumulating. That repression of FH is interesting. We linked this eventually into type 1 interferons, amazingly. And this was in our paper 1.5 years ago or so. If you repress FH in a macrophage, TCA cycle unravels. The mitochondria hate that. They don't like a lack of a TCA cycle. What do? To our surprise, they release RNA. Double-stranded RNA comes out, is sensed by RIG-I and MDA5. They're the well-known RNA sensors. MABS is in the mitochondrial membrane conveniently and drives type 1 interferon. There was a paper on this, FH repression, driving RNA release to drive interferons. And we were pleased with that paper because nobody linked those things together before. And here's some of the evidence for this. Here's the boost in interferons that you see with FH repression, right? If you knock down RIG I or MDA5, you'll lose that boost. In collaboration with Caroline's not here, is she? Caroline Jeffries and Cedars-Sinai. I wonder if she was here. She gave us loads of lupus data. In lupus samples, FH is repressed. Now remember, lupus is an interferonopathy. So one reason we think is FH repression, giving rise to interferons. And then more recently, Salmonella does the same thing. Salmonella represses FH very clearly in macrophages. If you block FH, you get a big increase in interferons with FH repression with Salmonella. And then 2 weeks ago, a disease called systemic sclerosis, they're getting repression of FH here as well. So there's loads of places then where FH repression equals mitochondrial disturbance, RNA release, and interferons. And so that's a new thing that's emerged. And then finally, in a type of renal cell carcinoma, where FH deficiency is a feature of this, again, they showed nucleic acid release through FH repression. That was Christian Freza's work. Now, lastly, though, our latest stuff. Now, so you get the message, I'll reiterate it for you, that was complicated. FH repression equals interferons through RNA release, right? Itaconate could block SDH. That's another enzyme in Krebs cycle. We wondered would itaconate affect interferons because it can block SDH. And in fact, it had been shown by Amanda Swain and colleagues that itaconate could boost interferon. And here we see itaconate driving interferons. Here we see ACOD1 knockout mice. They can't make interferon in response to LPS. We knocked it down in human monocytes. And again, you can repress interferon production by knocking down ACOD expression. Now, what this means is endogenous itaconate is required for interferon beta. Isn't that amazing? Because if you can't make itaconate, you cannot make interferons. And that's a strange link from a Krebs cycle byproduct into the interferon pathway. Again, RNA-seq, massive increase in interferons and not much else in response to itaconate. That was nice to confirm that. And then, of course, we had to prove it was SDH inhibition. Remember that itaconate blocks SDH. Could we show that's the mechanism? Here's bog standard SDH inhibitors, boosted interferon with a thing called AA5, TTFA, boosted interferon. We got some SDHA deficient macrophages, huge increase in interferon. Now, again, remember, this is all about disturbing Krebs cycle. And then a very good pediatric oncologist in Dublin called Maureen O'Sullivan, I met her at a conference. She works on SDH deficient tumors. They're called paraganglioma. Massive increase in interferons in those tumors as well. Again, linking SDH into this process. And we looked at RIG-I and MDA5, again, come up in the reactome analysis. And lastly, then, it is about RNA release. So itaconate will cause the mitochondria to spit their RNA out into the cytosol, but not in the RG1 knockout. So here we see the wild types, RNA release, knockout RG1, ACOD, no RNA in the cytosol. 4 different mitochondrial RNA species are here. So again, we could show the following was going on, and this summarizes it. Surprisingly, I mean, Krebs would be turning in his grave, wouldn't he? He knew about it. LPS represses FH. You got that already, right? Secondly, it induces ACOD and you make itaconate. That suppresses SDH. 2 key enzymes in Krebs cycle are now going down. They're going down. What happens is the mitochondria goes, I don't like this. And it releases. Its mitochondrial RNA gets released. MDA5 and RIG-I sense that, and now we get interferon production. So we've linked 2 enzymes in Krebs cycle to interferons is the way to think of it. And what this means overall for itaconate is 3 for the price of 1. It's antiviral. You're making interferons. It's immunomodulatory because it's controlling cytokines. And there was a literature back in the 70s, it can kill bacteria. It inhibits certain enzymes in bacteria. So a very intriguing molecule. I hope you agree. This molecule coming off Krebs cycle is intriguing. And it will have direct effects on certain viruses. And then there's the pro-tumor effect here. It's also got effects on obesity. So it's a really interesting metabolite. I show you this to illustrate. It's a single metabolite. There are thousands of metabolites changing here, and we don't know what they do. But this is one, and it's having all these different diverse effects. So to finish up, the 3 stories I told you. PKM2, adenosine, and IL-10. Complex 3 and IL-10 and itaconate. Let's look at the clinical aspects of this. Now, remember I told you autoimmunity is reaching epidemic levels. Remember that? Now, why is that? I think it's because of this. It's all about modern living is rubbish. Obesity, processed food, lack of exercise, circadian, pollution, all of that damages mitochondria. And I'm calling this a break-in endosymbiosis. Now, remember, these things are bacteria inside our cells after endosymbiosis. They're going rogue. They're reverting back to their bacterial origins and releasing all this stuff. DNA, RNA, metabolites, I've told you about ROS, all of these things are coming out, driving these diseases, and these go up because all of this goes up. So it's all about damage. And remember, IL-10, great mitoprotector, and that could be a way to stop this. And if you look at dysfunction of mitochondria, there's reviews on this. The green here is therapeutics. There's very few drugs have been approved that target mitochondria. This is publications. And yet there's an increase in evidence that mitochondria are key to all this. So again, I think this is very interesting. And going back to my notion, you want to turn that into that, right? Remember that? And of course, we know all PKM2 will do it. Many things can do this, though. Things like metformin, you know, MitoQ will do it. And now we think these PKM2 tetramerizers might prevent that breaking into symbiosis. That's one idea that we have. Second story, complex 3. Now, could you promote complex 3? That would be interesting. If you could block ROS, right, that would boost anti-tumor immunity because you'd be losing IL-10. Remember I mentioned IL-10 is pro-tumor. How would you do that? Well, there are efforts to block OXPHOS. Various drugs have been tried. There was a big trial done. Various molecules will block the electron transport chain. A big trial was done on the complex 1 inhibitor, very famous inhibitor called IXC 750. Trouble is, it's toxic. You're blocking the electron transport chain here. It's too dangerous, you know. However, maybe sequel 3 could be safer. If you block the ROS part of this, you might have a safer way to target the ETC in the oncology context. And then lastly, my favorite, itaconate. Now, obviously, I wouldn't be standing here telling you the whole story if we didn't have a drug in development. That was taken from my lab into Citrix, my company. Citrix, they would deal with Eli Lilly. That often happens, a big collaboration, a big drug company. Eli Lilly took the itaconate story and they made a molecule. They've made a drug based on 4OI. And wonderfully, it's now in clinical development. They've just finished. This is off the Lilly website. I want to mention Chuck Dorsey. He's an absolutely important collaborator inside Lilly. That's the champion. Need a champion. Luke Borner is the postdoc in Lilly. He did some of the stuff on interferon, actually, on RG1. But look at this. They have an itaconate mimetic. It's a chemical entity being studied for the treatment of autoimmune disease. They've finished the phase 1, and they're now starting in phase 2. They've picked one big autoimmune disease. They haven't disclosed what it is, and I can't, otherwise I'll be shot. But now we're in the clinic with an itaconate mimetic. Now you can see why Lilly would love this. First of all, DMF is there already and it mimics that, and that works in MS. Secondly, the steroid story is great. The exercise story, all the models where the itaconate derivative is anti-inflammatory, that gives a company confidence to pick a target. That trial will cost them $120 million, so you better be right, you know. If I get this wrong, that's the end of me. But we'll see. That mimetic is now about to start clinical trials, which is tremendous. And I think what the bottom line from all of this is, as I said earlier, 3 things in my lab then are now in the clinic, and we'll see if they work. Now remember, in case you don't get too excited, 80% failure right at this stage, 80% failures. Isn't that amazing? Why can't we get that better? I don't know. AI might help us, I suppose. So, but you can't be too excited yet. You got to wait and see. And just in case you're really bored, there's a paper here, itaconate keeps you awake. I'm considering making a coffee out of itaconate. This is a very, very strange paper here. Itaconate keeps you awake. It's very peculiar. So the 3 stories again. Remember, the tetramerizers of PKM2 could be for inflammation. Complex 3 derived ROS inhibitor for oncology. And now the itaconate derivatives with all of these diseases. And very lastly, I promise this is almost my last slide. Before all this, I worked in NLRP3. That was my big passion. That was the target. That's my old love. My new love became itaconate. And we made an inhibitor of that target, NLRP3. We were the first to find a small molecule inhibitor. It was called MCC 950 or CRID3. We had a company based on that called Inflazome. We were bought by Roche in 2020. They took our molecules. Roche, the biggest drug company in the world, have our molecules, and they're running 3 trials with an NLRP3 inhibitor, asthma, Parkinson's, and cardiovascular. And those trials are running as we speak with the inflammasome inhibitor that began in my lab. That's story number 4, I suppose. So we're very interested in that one. I guess I show you this because there's never been a better time, in my opinion, for inflammatory diseases, because all these compounds are in the clinic. Surely to God, one of them is going to work and slow down Alzheimer's or slow down Parkinson's or stop Crohn's. All these diseases are in the mix. So let's see what happens next. It's not for want of trying. Let's put it that way. And I'll finish there, and there's my lab. I mentioned all these people. Shane O'Carroll did all the work on itaconate and interferons. Alessia Zota did all the work on complex 3 that I mentioned for you. I mentioned Eva Paulson, Juliana rather. She was a key person there, and Eva Paulson. They were the key people in the IL-10 story. Martin Brandt gave us that molecule, very important. Mike Murphy, the collaborator, Caroline Jeffries, Acetylcyanide with the lupus study. Very important, Chuck and Luke and Lily, because they help us a huge amount. And Maureen gave us samples on the STH deficiencies that I told you about. And thank you very much for this.
I think we have time for a few questions.
Students, questions first.
Don't be scared, you students. I'll give you a Snickers bar. They're hungry.
Okay, I'll give you time. So just to close the loop further, I might have missed it because you're talking so slow. Final point in the loop, how does IL-10 re-elongate the mitochondria? And then one point in the very beginning of the loop, what is the functional benefit, functional role of the toxic state of the mitochondria? I actually like the toxic state of the mitochondria, but yeah.
No, they're good questions.
I like spicy food, you don't. So there you go.
Well, obviously this happens normally during inflammation, remember, as ever, inflammation or inflammatory diseases, it tips over into pathology, you know. So you need this to happen during infection. I mean, you do see mitochondrial fission, they make the rods to be, you know, activate the immune system. It's always a balance then, you know. When does it become pathologic? We presume it's just sustained activation, the toxic stuff in the environment, whatever it is. So there's some pivot into a damaged state, but we don't know is the answer. Of course, ultimately, I'm waffling away there. But there's some kind of event happening to make that persistent.
Which means that you propose an experiment to fragment mitochondria and see if this would activate inflammatory state.
Yeah, exactly. Yeah. Well, it's a bit causation, correlation to some extent, of course. But you will see mitochondrial fission during inflammation normally. So the question then becomes, how does it become pathologic? Now, the IL-10 bit is a great question. We don't know. IL-10 is somehow driving signals to maintain mitochondrial health. There's some evidence of STAT3 going to mitochondria, for instance, and going into the mitochondria, you know. That's a really interesting thing to figure out. And nobody's done a metabolomic analysis with IL-10, for instance. So just putting IL-10 in a macrophage actually will maintain the mitochondria. And again, that must be part of its anti-inflammatory immunomodulatory effect. But that's something that we've uncovered that needs further exploration to figure out what IL-10 is doing. It's a great one to think about.
Okay.
Okay, that was phenomenal. That was really great. But the interferon story caught my attention.
Yeah.
I think for two reasons. First of all, what about the TRIF axis with LPS? And the second then is, if atacanate is anti-inflammatory but driving interferon, there must also be some sort of cost balance thing going on there.
Yeah, well spotted. Both counts well spotted. Interferon, again, I'm going to wave my hands and waffle for a while. Interferons are interesting, remember? Someone that were interferon beta is used in MS, for instance, you know, so there's something going on with the interferon response here that's more immunomodulatory, you might say. And then in certain situations become pathologic. So you might have a situation where in lupus, you don't want itaconate to drive the interferon. If you could modulate that, that could be useful, you know. So that's one possibility. But I think it's to do with that interferon literature being so complex. The TRIF thing, well, it's all coming off TOL4 anyway. So we think much of this metabolic reprogramming is doing things like HIF activation, for instance, driving certain things. So it's coming off the TRIF-TRAM pathway. Whatever way you look at it, everything is initiated TOL4. I guess what we don't know is how TOL4 signaling represses OXPHOS.
Yeah. But
we don't know what the connection there really. We don't have that.
Right. But you had bar graphs indicating that ACOT is required.
Yeah,
for
yeah.
interferon beta expression in response to LPS.
Absolutely. So what's happening there is TLRs are driving interferons initially, right? They feed back and drive ACOT. ACOT makes itaconate to suppress SDH to cause the RNA release to soup it up. So it's a bit like a positive forward loop there. It's separate to the TRIF-TRAM getting into TBK1. But it involves that ultimately through the nucleic acids. And my other thing about the breaking into symbiosis, I mean, that idea is there already. So viruses, a good example is this, DNA viruses need RIG-I and MDA5 to drive interference. And when that was seen, what's going on here? These are DNA viruses. It should just be C-gas, you know. But it turns out they're driving RNA release from mitochondria to plug into RIG-I and MDA5. So in other words, that the mitochondria are acting like an accelerator, you might say, through their own nucleic acids. And in we did a review on this, which is I may recommend to you. It was in Nature last year. So there's very good evidence now that you get a PAMP-DAMP combination. So the PAMP is the viral nucleic acid. It could be DNA or RNA. The PAMP. The DAMP is the nucleic acids in the mitochondria, and both are needed. You got optimum interferon production. Do you know what I mean? So it's a really interesting development that I think. Now, in fact, that may be the best example of the breaking into symbiosis because the virus is doing it. Will things like processed food or pollutants break into symbiosis as well? And there is some evidence for that as well. So what's happening is there's a focus more and more on mitochondrial nucleic acids. And the strange thing is they're going through the receptors that were discovered by infectious disease people. Obviously, it was a virologist that found it, RIG-I, but now it's sensing your own RNA, so that whole aspect I think is really fascinating.
Thank you very much for these new insights in metabolism. So my question is the TEP. Obviously overcomes a rate limiting step from PEP to pyruvate.
Yeah.
Is that true? And why is that so, that this is a rate limiting step in biochemistry?
You're going back to the 1950s there now with rate limiting steps. I don't know. It is a rate limiting step in glycolysis for definite, but it's driven by the tetramerization, remember. That's a tetramer in that state, right? If that becomes a dimer, it's a lot less active and you slow down glycolysis because it's rate limiting. So, but that was well known back in the 70s and 80s when they were characterizing the control of those enzymes, Now this business of the dimer, leaving glycolysis and going to the nucleus, that's pretty wild because it's a separate function entirely, binding HIF and controlling HIF-dependent genes. And in fact, many enzymes in glycolysis, they call it moonlighting. It's a separate function from their roles in glycolysis. GAPDH is a great one. That binds RNA and controls translation. And GAPDH is a rate-limiting step as well in glycolysis. So it could well be that the rate-limiting enzymes are the ones that have a separate function. That could be part of this, really. So it's a good question to ask in terms of rate-limiting steps. They're rate limiting because you need NAD for GAPDH, for instance. There's reasons why they're rate limiting. The reason this is rate limiting is it's the dimer tetramer ratio that controls that step in glycolysis.
It also is valid probably for the TCA intermediates. If you have an accumulation of one, obviously it goes in wrong pathways.
You've got it. Absolutely. And itaconate's coming off a conatate, and that'll slow down Krebs, and then begins to block SDH to really limit Krebs. So it's all about those feed forward loops and negative feedback loops. Now the biochemist says, are you a biochemist originally? Well, the biochemist worked on this for decades, the rate limiting, the control of these metabolic pathways. Now they're relevant in this context, which makes it very exciting. And I'd love to revisit some of those other papers on control points to see how they plug into different processes. So it's a really good point. It's really interesting.
So you could model it in principle with the new power of the computers, right?
You've got it.
It was so difficult before.
Yeah.
But with if you know the
Alex needs to do this.
KD values, you can. Okay.
How they did it back in the 70s is beyond me. I mean, they were using rate constants for enzymes and all this kind of thing. It was called metabolic flux analysis back then, of course. But now we should absolutely reapply this technology to get more insights into it.
We have time for 2 more questions.
Thank you for it. Oh,
Might do 3. It took me 2 days to get here. You know what I mean? So they gotta make the most of it.
okay. Thank you for a very insightful talk. So I have 2 questions. First is, you showed that sequel 3 actually blocks tumor progression, right?
Yeah.
So, but at the same time, I think Jonathan Kagan published this paper that if you treat the you inject LPS at tumors,
Yeah.
they make the disease a super disease. So it kind of creates
Yeah.
a pro-inflammatory environment, right? And then you have a lot of, and that also causes tumor clearance. So you think it's kind of context dependent? That is first. And the second is, do you think itaconate is going to treat inflammaging as well and then just increase the healthy lifespan of people?
Yeah, second one first, absolutely. In fact, I was saying there this morning to say we got to look at itaconate in an aging context now because no one's looked at that yet. We'd love to know how it changes with aging, see what's going on. It'll be going down, you predict, and then you get more inflammation. That's well worth looking at. The tumor business, well, as you know, there's the double-edged sword, as they always say, because pro-inflammatory environment can be pro-tumor. Now, and sometimes anti-tumor, depending on the context. Yeah. In this case, though, certainly B16, I'm going to pick B16 because I'm IL-10 sensitive. It may not work in other situations. The other thing we found was the tumor cell itself, of course, will make IL-10. And we can repress IL-10 from the tumor by blocking complex 3 as well. So again, that's interesting from that point. It's early days, this stuff. We need to do more. It's great to see people do more work. We published a paper mainly to encourage people to try sequence. There's only like 3 papers on it out there. You know, so it's great to see it in different contexts.
Thank you for this. It was a very exciting talk. I had a question. Does IL-10 support mitochondrial health in non-immune cell types, or is this conserved to macrophages? And second part, do you see it as sort of an evolutionary mechanism to support symbiosis that's retained?
These are great questions, absolutely. Yeah, I go beyond macrophage, I bet you. That IL-10 is a myeloprotector in many cell types. We've no evidence for that, but I suspect it would work in other cell types. Worth looking at though. I mean, what's happening a bit is we publish in macrophages, then someone else tries the epithelium and the endothelium. There are differences, of course, but there can be similarities. So we're considering that. In terms of endosymbiosis, that's a great question to ask. The biggest mystery of all is how that happened, remember. In other words, how did we retain the endosymbionts and not eat it, you know? So maybe there was some primitive IL-10 like pathway that was allowing that in the symbiont to survive. That's a really good question. But we have found recently, interestingly, you know, I gave this talk, similar talk about 4 months ago, in UMass, right? And there was some bioinformatics guy in the audience who works on the evolution of all these receptors. And he said to me, he went back to his lab, you know, and he said MDA5 looks like it evolved at the time of endosymbiosis. Like the data is there. So maybe that evolved, since the RNA, somehow maintained homeostasis. This kind of thing. You know, so it does, you know, speaks to that whole notion of evolution and how endosymbiosis occurred in some ways. So it's really interesting that part.
I think we have one more question back there from Lena. Is our bonus question? So.
Great talks, plural. My question was about the connection between IL-10 and mitochondria. So you show that IL-10 promotes OXPHOS, but then reactive oxygen species from complex 3 is required and promotes IL-10 production. So wouldn't that lead to a feed-forward loop that's just bad for mitochondria?
A feed-forward loop.
Where you're just getting more ROS, more OXPHOS, more ROS, more OXPHOS.
Yeah, possibly. But remember, there's great antioxidant systems to mop up the ROS that are probably there as well. Is one problem, you know.
Does IL-10 induce any antioxidant?
I think it does. I think it induces heme oxygenase, for instance. But it's these are the kinds of things we need to revisit IL-10, actually, I think, you know, in this regard, to see exactly what it's doing metabolically to cells in terms of antioxidant responses. Feed forward, of course, is in general anti-inflammatory anyway, because you're coupling OXPHOS. You're getting less ROS overall than you would if you begin to block some of these things, you know. So overall, it's seen as a net anti-inflammatory process, I would think. A big reason for that, of course, is the membrane potential is being controlled properly. If you get hyperpolarization of the mitochondrial membrane, the mitochondria begin to release their nucleic acids, which becomes a net pro-inflammatory effect. Do you know what I mean? So if IL-10 can couple OXPHOS, shall we say, that would be a net anti-inflammatory process, is the idea. Now, the question then is, how does LPS boost the complex 3 ROS connection? That's something that we don't know. So why would LPS? We know we published a paper in 2016 actually on reverse electron transport through complex 1 driving ROS with LPS. That was a strange business. Electrons flow backwards in that situation. But how complex 3 is being regulated is our next question. We'd love to find out with LPS as a trigger. We'd love to know more about that.
Thank you, Dr. O'Neill, for.
Thank you very much.
Good seminar.